All right, thank you. As systems biologists, we're really interested in understanding the combinatorial interactions in cells that really then lead to organismal properties. So phenotypes, traits, and of course, disease states. So we use a lot of data analytics and now increasingly explainable AI algorithms and supercomputing context to find the combinatorics, the interactions within and across all omics layers. So whether that's genome or epigenome or transcriptome or proteome, metabolome, microbiome, how all of that works together to impact phenome. And then of course, how all of that is conditional on environment. So typically we try to capture those relationships and often use network models to capture those relationships. So the nodes in a network can represent really any biological object, whether that's a gene or a genome variant, a methylation event, a transcript, you name it. You can represent that as a node in a network, and the edges are simply the relationships between those nodes. And you can have mixtures of different types of nodes, and you can have mixtures of different types of edges, whether those are physical connection edges in a protein-protein interaction context or a statistical association in an edge. Again, you can have whatever relationships you would like, you can model in a network. We're of course trying to understand the fundamental biology of what's going on in this case in SARS-CoV-2 is the virus, as well as the pathogenesis in response in humans themselves. In using these types of networks, we write a lot of different network mining algorithms to try to find the biological narratives, the functional mechanisms that are captured in those network representations of a complex system. We're also, of course, interested in the three-dimensional interaction space of what's going on in cells. And so we've spent quite a bit of effort and have some brand new workflows or pipelines in my lab up and running on the Summit supercomputer that allows us to predict the three-dimensional structure of the majority of the proteins in a proteome for a species, predicted from primary sequence. This is a new pipeline, and it's an ensemble method. It uses often multiple different prediction methods per protein. The first step is to analyze the sequence of the protein and identify what different methods will be most beneficial on different parts of the protein itself. And by ensemble modeling, we can get a pretty good structural model for most proteins. There are some disordered proteins that don't have structure before they bind that we can't address, but we can get pretty good models. So we compare those routinely to existing crystal structures as sort of a testing phenomena, hold the crystal structures out and then predict the structure and compare them, and the models are often pretty decent. Now we're interested in that, of course, for understanding interactions between proteins and from a molecular evolution point of view, the effect of mutations. We're also, of course, interested on small molecule interactions, existing drugs in our case, and how they may stabilize or destabilize proteins of interest. So with that as a framework, we're attacking the COVID-19 pandemic from several different angles. One is trying to learn more about the life cycle and lifestyle of coronaviruses in general and SARS-CoV-2 in particular. So in a paper that just came out recently, we did a sort of molecular evolution comparison of SARS-CoV-2 to past coronaviruses, other coronaviruses, and their different, and what's similar between them and what's different between them, and how in three-dimensional space those mutational differences will impact function and pathogenicity. One of the outcomes of that study was the insight that one of the main proteases in the virus, 3CL-Pro, actually interferes with NF-kappa B. It actually looks like it cleaves NEMO, which is a key regulator of NF-kappa B. And NF-kappa B has some interesting sets of genes that it regulates, both in the Renin-angiotensin system as well as in the immune system. So hang on to that, that'll be relevant in just a second. We've been staring at the Renin-angiotensin system for a while, partially from the molecular evolution studies, with the realization that multiple mammalian coronaviruses get into the host cells via different elements of the renin-angiotensin system. So of course, we've all heard a lot about SARS-CoV-2 and its use of the ACE2 protein, which is a cell surface protease. But if you look around at other viruses like MERS or TGEV, you'll find that they're using other elements of these pathways to get into cells. So we were sort of preconditioned to be looking at the RAS pathways. And in this simplified diagram, you can see there's this really dynamic tension between ACE2 and ACE. Now, normally renin, the RAS system is really regulating blood pressure and fluid homeostasis. Many of you are probably familiar with ACE by the use of ACE inhibitors as one of the primary pharmaceutical approaches to hypertension. If you inhibit ACE and leave ACE2 alone or increase ACE2, you're going to tip the balance in the pathway away from angiotensin 2 and towards angiotensin 1-9. That will have some consequences, as angiotensin 1-9 specifically binds to the ATGR2 receptor. That binding event actually resensitizes the bradykinin receptors, and we'll come to that in just a second. So one of the reasons this is relevant is NF-kappa B, which, as we mentioned, seems to be dysregulated, is a positive regulator of ACE expression. So dysregulating NF-kappa B, you would expect to see a decrease in ACE expression. So with that framework in mind, we're looking at lots and lots of different types of data sets for these studies. So we're using lots of omics data. Today I'll talk a lot about gene expression data. So one of the data sets is a population scale data set of about 17,000 different RNA-seq data sets across 57 different human tissues, across about 1,300 different individuals. So with this in mind, and those are all uninfected individuals. So with a focus on the renin-angiotensin system initially, we wanted to see how are things normally regulated? What are the normal regulatory circuits? And what else should we be aware of in looking at the RAS pathways? So this figures the result of one of those analyses where we pumped those large population scale data sets through a range of both data analytic and now our new Explainable-AI algorithms to try to find what's going on with some of these key genes in the renin-angiotensin pathways. And here we see the sort of, again, the dynamic tension, the bilateral access between ACE and ACE2 and the AGTR1 and AGTR2 receptors. We start to see indications of the kallikrein-kinin system being involved in bradykinin. We also start to see relationships in parts of the immune system, especially inflammatory responses. So this gave us a framework to understand how things are normally regulated. And next, we wanted to look at what's going on specifically in the lungs of COVID-19 patients. So here we're looking at bronchoalveolar lavage samples. So very simply, you're wedging an endoscope down into the terminal bronchi of the lungs and injecting saline and sucking it back out. And if you do that about 15 times, you get a good wash of the alveoli surfaces, where of course you're normally doing gas exchange. So of the 50 to 100 square meters of surface area inside your lungs, that's heavily lined with capillaries for gas exchange. So we're trying to get a sampling here of cells that are in that key environment where gas exchange is happening, and you'll get a collection of epithelial cells as well as in lots of immune cells that are located in those environments normally. And we're able to do this for COVID-19 patients as well as comparing them to uninfected controls. When we look at the results of that differential expression results in the context of the renin-angiotensin system as well as the kallikrein-kinin system, we see some really interesting patterns that start to emerge. So the renin-angiotensin system, one of the very first steps in it is actually modulated by the vitamin D receptor. So renin converts angiotensinogen into angiotensin 1. the vitamin D receptor is a negative regulator. It's a repressor of renin expression. Well, what we see in COVID-19 patients is a severe upregulation of two genes responsible for catabolizing vitamin D and its metabolites. We see a significant downregulation in the vitamin D receptor itself. When we look in clinical records where vitamin D has been measured, we do in fact often see low levels of vitamin D in severe COVID-19 patients. From that interaction, you would expect to see an increase in renin expression, and that's exactly what we see, is an overexpression of renin. So this is sort of opening the floodgates into the RAS pathways. And here is where we see this balance sort of tipping with the significant downregulation of 8. This is an 8-fold downregulation of ACE. So you're going to tip the balance away from angiotensin 2 towards angiotensin 1-9, and as I mentioned previously, that will bind to AGTR2 and start resensitizing the bradykinin receptors. So normally the bradykinin receptors, when they see too much bradykinin, they desensitize, they downregulate themselves. Well, this forces them to keep listening to that signal. If now we go to the very top of the kallikrein-kinin system, we see that SERPING1 gene encodes actually the C1 inhibitor protein, which plays a key role in several different pathways. But here we're taking the context of the kallikrein-kinin system. It's normally a repressor of the top of that pathway. So it's significantly downregulated, again, sort of opening the floodgates into the kallikrein-kinin system. We see several of the genes involved in proteases involved in the kallikrein-kinin system upregulated. KLKB1 is neutral. These two are up, and that will lead to an excess of bradykinin, and CPN1 that is responsible for converting bradykinin to an alternative active form, desarch bradykinin, is significantly upregulated as well. Now, ACE2 comes into play here as well. It plays a role in the kallikrein system as it's primarily responsible for degrading bradykinin. It's heavily expressed in lung tissue, and in fact, it's in lungs that is probably the central clearinghouse for bradykinin in the system. So again, the downregulation of ACE, I mean, we're not degrading this excess of bradykinin. The two receptors for bradykinin are significantly upregulated themselves. Once you bind to a bradykinin receptor, one of the things that happens is you activate phospholipase A2, which kicks off the arachidonic acid cascade, leading into the prostaglandin signaling pathways. The result of that, of excess bradykinin, we know from other conditions, is hyperpermeability. So you'll become hypotensive, and you'll start to lose fluid homeostasis where this is happening. So in opening up gaps in those capillaries on the internal surface of the lung, you're going to have plasma fluid start leaking into the alveoli. Having fluid pouring into your lungs is never a good thing. Also concomitant with that, you're allowing immune cells normally constrained to the bloodstream to also squeeze into the surrounding tissue. So you're seeing the inflammatory response. The prostaglandin cascade also leads to chemotaxis to further attract immune cells into the area and further the immune response and downstream involvement with cytokines. Now, if we track this further, we also know that increase in prostaglandins should lead to an increase in the expression of hyaluronic acid synthases. And in fact, we see exactly that in the data. All three hyaluronic acid synthases are significantly upregulated. The downregulation of ACE, we would predict, would lead to a decrease in expression of the two genes most responsible for the degradation of hyaluronic acid, and that's in fact what we do see, a significant downregulation of those genes. Now, hyaluronic acid is this cool biopolymer that shows up all throughout biology. Bacteria produce it, so that sliminess of a biofilm or bacterial culture, a lot of that is hyaluronic acid. Snails produce it, that slimy snail trail, a lot of that is hyaluronic acid. Now, we as humans produce it in multiple places in our bodies. It's used as effectively a lubricant in joints. There's a feedback loop between bradykinin and hyaluronic acid. Hyaluronic acid actually has analgesic effects on the pain sensitization caused by one of the bradykinin receptors. And it's often normally expressed in low levels in the lungs, where it forms a thin protective layer and serves as a cell surface lubricant to help you move mucus across the respiratory tract. The bad news is it's also a very potent hydrogel. It can absorb more than 1,000 times its own weight in water. So in this instance where you're leaking fluid into your lungs and you're overproducing hyaluronic acid, you would expect to see a buildup of hydrogel. And in the autopsy reports, that's exactly what we see. The lungs of COVID-19 patients weigh twice as much as a normal healthy lung. And the pathologists talk about it feeling like a water balloon full of jello. And we can see in the histology slides from COVID-19 patients from autopsies that there's this layer of hyaluronic acid, this hydrogel built up in the alveoli. So you can imagine that that makes gas exchange of those surfaces very difficult. The incoming air has a very hard time diffusing through, and the CO2 has a hard time getting out. So this helps explain these sort of horrific mortality statistics we see in the ICU. Once you've intubated and ventilated patients, you have pretty bad outcomes. And a lot of that may well be because no matter how much oxygen you're pumping in, you just can't use to this hydrogel. I'm sure you've all heard about the radiological findings in COVID-19 patients of ground glass opacities. Well, in patients from other conditions where they overproduce hyaluronic acid in the lungs, we see exactly these same radiological findings, these white spots, these ground glass opacities. So I think the radiological findings in COVID-19 patients, a lot of this is the hyaluronic acid. Now, alarmingly, several weeks ago, a paper came out looking at asymptomatic patients. I'm doing chest x-rays on them and finding in roughly half of asymptomatic patients, they also have these ground glass opacities, where you simply don't see them in uninfected individuals. So that's an indication that damage is being done even though people aren't perceiving it. We're doing a lot of work with long hauler communities, and many of them report very mild or even asymptomatic acute disease phases, although they tested positive. But long term, they develop a series of quite debilitating symptoms that persist. From a public health perspective, this has sort of profound implications of how we should think about the spread of this virus. Even if people aren't showing symptoms, this damage is being done, and if a significant portion of them go on to have long-term impacts and something akin to disabilities, that has a profound societal impact that we need to be really concerned about. So we're busily collecting data and working with these groups and starting to view them really as a potential distributed cohort to investigate these long-term issues. If we project this as really a local model of infection, so local pathogenesis, and from the gene expression studies, we can look at expression patterns of the Renin-angiotensin system, and especially ACE2, where it's distributed in tissues. We can predict where what tissues viruses are likely to, this virus is likely to colonize. And autopsy studies back up the considerable tropism of this virus. It can colonize lots of different organ systems and tissues. Looking at those that you would predict it and where it's been shown in autopsies to be able to colonize and the symptoms associated with that, well, if there's an excess of bradykinin in muscle and joint tissue, we know from other conditions and animal studies that will lead to considerable myalgia, so really the sore, achy muscles and joints. An excess of bradykinin in the brain will lead to dizziness and headaches, cognitive impairments. We hear a lot about brain fog from COVID-19 patients, and indeed fluid-based ischemia. MRI studies of COVID-19 patients are showing the sort of leakage of fluid and swelling of the brain, the pulmonary edema and hydrogel I talked about in the lungs. If this is happening in the intestines, you get an excess of bradykinin will cause fluid to flow into the lumen of the intestine. Well, that's diarrhea, and the surrounding cramping and pain sensitization from the other bradykinin receptor, which is these sorts of GI symptoms are very common in COVID-19 patients. You would expect to see considerable levels of hypotension in severely ill COVID-19 patients from this model, and in fact we do. If this is happening in the heart, and it's clear there is heart involvement in COVID-19, that will cause big shifts in potassium. So shifts in electrolytes like that is known to lead to arrhythmia and sudden heart failure, which is also things we see in COVID-19 patients. So as we sort of march across much of the symptomology we see in COVID-19 patients, it matches up quite well to this model. We have other papers either under review or in prep looking at the cross connects between this model of a bradykinin storm and coagulopathy, and we can see some very bizarre things going on in the coagulation and fibrinolytic pathways, as well as the complement pathways. There is crosstalk across these systems, so we're rolling those out as we expand this model laterally as well as downstream into the signaling cascades. Now, that's all the bad news. I think the good news is there are already existing pharmaceuticals that hit a lot of the key points of this model. So, and several are being tried in the clinic. So there's a good study out of Spain using calciferodiol, which is a vitamin D analog, showing good efficacy in hospitalized COVID-19 patients. Relatively small study, needs to be scaled up, and we're working with folks to scale that up. There's another small clinical study out of the Netherlands with icabinate, bradykinin receptor inhibitor, also showing encouraging results in COVID-19 patients. And of course, we've all heard a lot about dexamethasone, either from the 7 academic studies and the meta-analysis from the WHO showing efficacy, or from the recent White House news. But dexamethasone seems to be effective, and it's inhibiting phospholipase A2, the first protein activated by the bradykinin receptors. As we've looked downstream, we found other potential genes that, other potential drugs that will interfere with the downstream signaling cascades of this mechanism. We've actually looked in the clinical data, and they would tend to confirm the model as well. So we have several different points in this model already. We're getting early clinical indicators supporting the model. There are other things that need to be tried. There's drugs that can affect the lack of the C1 inhibitor. There's a monoclonal antibody to KLKB1. There are drugs that can inhibit hyaluronic acid synthases. And in cancer therapies, hyaluronic acid is sometimes an issue in cancer, and so there's have been successful therapeutic uses of hyaluronidases to degrade hyaluronic acid. We're working really hard to push large-scale, properly designed clinical trials of these, and we're really interested in combinatorial therapies. We think it's probably going to be wise to hit multiple parts of this mechanistic model all at the same time to really shut it down. But the popular analogy for that is if you sail your sailboat over a reef and punch 5 holes in the bottom of your boat, you hope you have more than one cork. You'd like to be able to plug more than one of the holes. So we're pushing hard on this and looking for academic medical center collaborators to spool up larger and larger clinical trials. Some of this work was published in a paper in eLife earlier this summer. And we're also doing a lot of mining of clinical data. So we're fortunate at Oak Ridge to have the full electronic health records for 23 million people going back 20 years. And we have nightly updates of all those electronic health records. We are, of course, now tracking all the COVID-19 patients in that population and mining the clinical records with a variety of different phenotypic outcomes for COVID-19 to try to understand associations. So now we're using these Explainable-AI algorithms across clinical information and demographic information to try to find connections within and across these layers that are predictive of COVID-19 phenotypes and outcomes. And we're already finding fascinating insights both into comorbidities and some surprising comorbidities we wouldn't have thought of without these Explainable-AI approaches. We're also using more hypothesis-driven clinical epidemiology approaches to this, and a couple papers under review on that already. But these are also, we're also finding interesting insights into drugs that people happen to already be on, and some of which are appear to be protective against bad COVID-19 outcomes, many of which already fit into our model. We have clinical data supporting other elements of the model as well. We're mining, we're part of the COVID-19 host genetics initiative, so we're bringing the genetic signal into our models as well. And as I mentioned, a lot of molecular evolution studies on the virus itself, trying to get inferences into viral biology and function, as well as not just how it compares to previous coronaviruses, but how it's changing during the course of the pandemic. And we just got, hopefully we're in the very final stages of review on a very large scale study on how it's changing during the course of the pandemic. We just received funding to start looking at the zoonosis side of this and predicting and preventing future pandemics. Very briefly, I want to just touch on the environmental side of this.
A minute or so, Daniel. Yeah.
Sure, can do. So in addition to the omics and clinical data, we're really interested in understanding the environmental conditionality of this. So we happen to have, for other reasons, we are very interested in environmental signatures. So we've constructed a data set of both environmental information as well as socio-demographic information, socioeconomic information, policy information. The environmental information we have for every square kilometer of land on the planet. So we represent that information in vectors with about 414,000 elements in them. We've used the Explainable-AI models to look at the environmental signatures and see if there are associations with COVID-19 outcomes, mortality, and in fact see pretty strong signatures of impacts on environment on COVID-19 outcomes. So we want to understand these environmental patterns globally. So we've done all against all comparisons of those 156 million vectors. To do that, we had to hit exascale speed to pull it off. This turns out to be a 22 zeta op calculation. It's 10 to the 22 mathematical operations. That would have taken over 100 years on my laptop on this 170 terabyte data set. It took us 3 hours on the Summit supercomputer. To our knowledge, this is the largest calculation done in human history. We can now represent the results of that calculation as a network, and now we have representations of the similarity of all points on Earth. One uses of that is we can simply cluster that network. As we increase the granularity of that clustering, we can go from these very broad traditional climatic representations into finer and finer grain patterns as we crank up the granularity. So that now we have relationships across the entire globe. And I can pick any point in the planet and show you what it's like. So this region in northern Australia, for example, if we follow across, we find that in Mozambique. We find very similar environments in southwestern Africa and further south in South America. We can do this for every environment everywhere on the planet. So this has lots of applications, not just to infectious disease, but other clinical conditions that are conditional on environment. Of course, we're using this also in our bioenergy and precision agriculture studies and microbiomes and ecosystem studies. We're able to do a lot of this work because of this sort of extreme compute power we have at Oak Ridge. Shout outs to our various funders and new funders coming in. We have a new round of NIH money and new rounds from other agencies. My fantastic co-authors, both at Oak Ridge as well as at the VA, Yale, Cincinnati, and Wisconsin, and this phenomenal group of postdocs and PhD students, staff and faculty members that I'm so fortunate to lead. And with that, we can take questions.
All right, thank you very much for that fascinating talk. There's obviously a lot more we could say on all of that. I appreciate that. You're getting to wrap up, and the networks that are being built are, incredibly interactively complicated in the biochemistry of it. So one question comes to me right away is, where and how many points does the virus initiate all those network signals? So you mentioned cleavage of NEMO. I saw a couple of CYP3A4 and others up at the top of some of those. So do we know how many points the virus is entering into those networks?
We're looking very hard at that. I mean, it's amazing the havoc that this little bag of 27 or so proteins can cause. So far, we're aware of over 300 different viral to human protein interactions, and a similar number of viral RNA to human protein interactions. And so there's a lot of things going on. We're very busily incorporating that protein interaction data into our network models as well, and mining those other parts of that. Some of those obviously are affecting the immune system. Others of them are involved in, as the virus takes over a cell, it very cleverly shuts down the translation of host transcripts, and biases towards translation of viral transcripts. I mean, just fascinating mechanisms. There's a lot of that at play that we, and of course many others, are all trying to tease out. We're trying to look at this holistically of how this impacts these sort of pathogenesis models and immune models and coagulopathy models, and fibrin deposition models. But yeah, it's an amazing multitasker of a little beast.
Right. So, I mean, your analogy of plugging all the holes in the leaking boat seems right in the sense that, and because there are so many holes, it's going to be difficult. And the higher up the chain you can find to stop this will be better, presumably. Yeah.
Right. We're also actively involved in sort of, in the DOE system, there's a cross-national lab organization called the National Virtual Biotechnology Laboratory, all the national labs working together. And a lot of that work is identifying these sorts of targets and then finding inhibitors for viral proteins to try to shut down some of the viral interactions right at the source.